Hello students, a very warm welcome to my online lecture. Today we gonna see a very important clinical question which was asked in 2019 university exam of SRM University Medical College. It's a very university. I still remember that this question was not answered by the students. 90% of the students could not answer this question because it was unexpected from carbohydrate metabolism. In a initial starting step itself, I am saying this clinical question is from carbohydrate metabolism. Carb metabolism. Remember carb metabolism. So now, without any further delay, we'll go through the question, clinical question. A male baby, let me take a highlighter so that I can highlight some important points in a clinical question. A male baby of 2 months age was brought to the pediatric OPD. Here, of course, 2 months baby means mostly pediatric OPD only admission. So let me highlight here a male baby of 2 months age, very little stage. So with a history of one episode of seizure, and the main complications here you have to highlight: yellowish discoloration of skin. Where can you see, when can you see yellowish discoloration of skin? Mostly in jaundice. Write it down: yellowish discoloration of skin, the first point. And in whom you saw this? Like baby. For the last 1 month with on and off fever, poor feeding. See here, feeding, poor feeding. Let me take a highlighter. Poor feeding, excessive crying after feeds, which means after taking milk, after fed by the mother, and presence of whitish discoloration within the eyes. Colored whitish discoloration within the eyes on presence of bilateral cataract. Cataract means it is a eye disorder. Okay, and hepatomegaly. Hepatomegaly means enlargement of liver. So what all complications you have seen in the baby? Male baby of 2 months, a very little kid, 2 months, developed yellow discoloration of skin, poor feeding, excessive crying after feeds, and whitish discoloration within the eyes. It is due to bilateral cataract, and hepatomegaly, enlargement of liver. So you see the complications. So after the tests were done, like biochemical tests were done after taking a blood sample from this male baby of 2 months, and once the tests were done, here are the results. Number second, here reducing sugar in urine was tested, and reducing sugar in urine gave us positive, which means there is a sugar in the baby's urine. So after Benedict's test, the result is positive and reducing sugar is present in that urine. And liver function test, here are the following liver function tests. Here you can see total bilirubin 20 mg per dl. Here we have normal reference values I have given. You can see here, you can check the normal reference values against the abnormal values. Here you can see here the baby has some a disorder is there in baby. So all these values that we got it from baby sample after the tests were done. So total bilirubin 20 mg per dl. The normal is bilirubin total normal is 0.2 to 1 mg. See here how minute the value is, the normal difference, but here 20 mg, which is very high. Direct bilirubin 6 mg, the normal is 0.4, less than 0.4. And indirect bilirubin here we got was 14 mg per dl, and here the normal is less than 0.6 mg per dl. Similarly, total protein, albumin, globulin, all they are very low, because what here we discussed in the clinical question, hepatomegaly, which means liver enlargement. There is a liver disorder, liver is defected, so albumin synthesis, globulin synthesis, protein synthesis will be taken place in the liver. So when the liver is not functioning, what happens? Automatically whatever products that we are getting from liver will be slowed down. So albumin level is very low, globulin low, AG ratio is abnormal, and AST, ALT, ALP, all these are liver enzymes. Whenever liver is defected, these enzymes will be elevated. Whenever liver is healthy, these enzymes will be in a normal ranges. You can see here AST range 10 to 50, and here ALT 10 to 45, and ALP 50 to 100. You can see here ALP is elevated. ALP means what? ALP normal value is 50 to 100 international units per liter. So but here you can see 200, so double times higher. So ALP will be elevated in bone disorders and also biliary apparatus. So ALP is increased, AST and ALT both are normal. So now you can see according to these values you can easily tell that there is a defect in liver due to what happens. There is a yellow discoloration of skin. Defect of liver leads to bilirubin. Generally if there is a jaundice, yellow discoloration of skin means jaundice only. So you can see yellow discoloration of skin or eye icterus and all that. What you say, if you see all that complications, you say jaundice. Jaundice is mainly related to the liver only, because bilirubin metabolism conjugation occurs in the liver only. So here you will before going to clear all these 4 questions, clinical questions under the under the main clinical question, we have sub questions. We have to answer these 4. So we understood now the normal values, which are these are normal reference values, and these are the values that we got it from a patient sample, the baby sample. And we also appreciated the complications of the baby. Now we gonna see 2 metabolic pathways in order to understand this whole clinical question clearly. So now we have here 2 metabolic pathways. In a simple way I'll explain. I don't take much time. If I take much time, you'll get bored and you will not listen. So remember here, these are very important metabolic pathways in carbohydrate metabolism. You must have to understand it. Yeah, you can see here. This is a galactose metabolism, which you can see in carbohydrate metabolism. Very, very important galactose metabolism. Actually, what I said in a clinical question, 2 months baby. 2 months baby means what is the food for 2 months baby? What they usually take? Only mother's milk. Mother's milk means what they'll get from mother's milk? Lactose. Okay, lactose is only the source for the newborn babies, like till sixth month. So now lactose, once lactose is fed by the baby through the milk, what are the sugars present in lactose? The main sugar is galactose. Of course, this lactose is a disaccharide. Now we only concentrate on galactose. So now here, of course, one other sugar is glucose, galactose. Once they had milk, mother's milk, the galactose will be produced. This galactose, after absorption, what happens? It enters into the liver. Galactose metabolism mostly occurs in the liver. Now this galactose entered into the liver cell. In the liver cell, this whole metabolism is occurring. This galactose is, as soon as galactose enters into the liver cell, this all occurs in the liver cell cytosol. Okay, cytosol. Liver cell cytosol. Galactose will be converted to galactose 1-phosphate by the enzyme galactokinase. Here ATP is required. This phosphate will be transferred to the galactose, and galactose is converted to galactose 1-phosphate. The formed galactose 1-phosphate will be attached with UDP glucose for a transfer of this UDP to this galactose. What happens once both are connected, galactose 1-phosphate and UDP glucose, both condense, and you will get, at a result you will get UDP galactose and glucose 1-phosphate. UDP galactose and glucose 1-phosphate. Galactose 1-phosphate takes up this UDP and gives you UDP galactose. And this glucose will take this phosphate and convert it to glucose 1-phosphate. It's like an exchange reaction from glucose 1-phosphate to UDP glucose to galactose 1-phosphate. Once UDP galactose is formed, this UDP galactose is a source for lactose in mammary gland, and also UDP galactose is required for the synthesis of all these glycosaminoglycans, glycolipids, glycoprotein synthesis and all, it is required. And glucose 1-phosphate, which is formed by the galactose, glucose, will be enter into the glycolysis, glucose 6-phosphate glycolysis and synthesis of glucose also. So of course all these are not needed. See here, the important one is this enzyme. Due to this enzyme deficiency, the child there, like the newborn baby, 2 months old baby, developed a jaundice and all the altered enzyme, like hepatic enzymes. You can see here galactose 1 phosphate and UDP galactose. Conversion of these two into these two requires this enzyme, galactose 1 phosphate uridyl transferase. What happens in babies? This enzyme will be defected. If this enzyme is defected, what happens? This galactose will produce galactose 1 phosphate. Galactose will produce galactose 1 phosphate. But this galactose 1 phosphate cannot be converted to UDP galactose and also automatically glucose 1 phosphate. What happens if this enzyme is deficient? This galactose 1 phosphate will be elevated. Where? In a liver. In a liver, this galactose 1 phosphate is elevated. Sometimes what happens? Galactokinase deficient, not galactose 1 phosphate uridyl transferase. In the deficiency of galactokinase, this galactose will not be converted to galactose 1 phosphate because there is no one to add phosphate to the galactose. This galactose will be elevated. This elevated galactose will go to the eyes and forms a galactose by aldose reductase. This accumulated galactose in blood, for example, if galactose is elevated in blood, that is called galactosemia. If galactose is present in urine, that is galactosuria. So these elevated levels of galactose in the deficiency of galactokinase, what happens is galactose will enter into the eye, in the retina, you can see here, and forms galactitol. Galactitol is an alcohol form of galactose which leads to cataract. So accumulation of galactitol or production of galactitol in the eyes leads to cataract. We have already seen in the question, baby has a defect in the eyes also. Here you can see bilateral cataract. So that bilateral cataract is due to what? This enzyme deficiency. Let me scroll it up. Yeah, galactokinase deficiency leads to galactitol, and galactose 1 phosphate uridyl transferase deficiency also leads to galactitol, because galactose 1 phosphate and galactose both will be elevated. Now we had already discussed there, the baby has a jaundice, like yellow discoloration of skin. We'll see why yellow discoloration of skin. Remember, this galactose 1 phosphate elevation due to the deficiency of galactose 1 phosphate uridyl transferase. So this is an enzyme. Reason of this, due to this enzyme deficiency, galactose 1 phosphate is elevated, and this elevated galactose 1 phosphate causes jaundice. We'll explain you how this elevated galactose 1 phosphate causes jaundice. For that, we need to see one other pathway, which is degradation of heme. We'll explain in a simple way. Here you can see aged erythrocytes, which means RBCs. After 120 days, they'll be engulfed by the macrophage. Here we have macrophage. Aged erythrocytes, RBC has a lifespan which is 120. After 120 days of RBC lifespan, these aged erythrocytes or RBCs will be engulfed by the macrophage in a spleen. So here hemoglobin is there. In RBC, what is present? Hemoglobin is present. Hemoglobin will be broken down into two pieces. For example, if you split a hemoglobin, what all you will get? You will get globin part and also heme part. Now we have heme part here and globin part is removed. We have only heme here. This heme will be converted to biliverdin by heme oxygenase. Biliverdin is converted to bilirubin by biliverdin reductase. This bilirubin is now in the macrophage. This bilirubin is a waste product and also harmful product. This bilirubin has to be flushed out from our body, but this is present in macrophage. If you want to eliminate something from our body, we have two routes. One is urine and the second one is stool. If you want to send this bilirubin out from our body, we have two routes, but this is in macrophage. First, bilirubin has to come inside the blood in order to go out from our body through urine. So for that reason, bilirubin enters into the blood. Since bilirubin is insoluble in blood, bilirubin is insoluble in blood, bilirubin takes help from albumin. So bilirubin takes help from albumin. Albumin is a protein. As I already told you, albumin is a protein made by the liver. So albumin helps this bilirubin to transport throughout the blood into the liver. This albumin takes up this bilirubin and delivered this bilirubin into the liver. Now this bilirubin, which was formed in the macrophage, is now in the liver. Bilirubin is reached here. For what? Bilirubin, I told you, insoluble. To convert soluble bilirubin, or insoluble is nothing but conjugated or direct. You can also call it as direct, conjugated, or insoluble. Soluble bilirubin is nothing but direct insoluble bilirubin is unconjugated. Soluble bilirubin is conjugated. To convert the stage of insoluble to soluble, bilirubin has reached the liver. In the blood, bilirubin is present in the form of conjugation with albumin. Here, liver, bilirubin is insoluble and ready to get converted to soluble form, which is bilirubin diglucuronide. You can see here, in order to convert this insoluble form of bilirubin to soluble form of bilirubin diglucuronide, what we need is bilirubin glucuronyl transferase. It's an enzyme, very important enzyme. What this enzyme does is, this enzyme converts bilirubin this into this, which means this insoluble is converted to soluble, this insoluble is converted to soluble, so that this soluble compound easily can enter into the intestine and kidney via these two routes. It can be easily excreted. Now you can see 2-UDP glucuronate. Here is a main point. If you understand this point, you can easily answer to this question, clinical question. 2-UDP glucuronate. UDP means uridyl diphosphate. Okay, uridine diphosphate, not uridyl, sorry, uridine diphosphate. Uridine diphosphate, 2-uridine diphosphate glucuronate. This glucuronate, which is connected to, which is bound form to 2-UDP compound, will be donated to the bilirubin. As soon as glucuronate is shifted to bilirubin or donated to bilirubin, this bilirubin utilizes this glucuronate and converted to bilirubin diglucuronide. Once bilirubin diglucuronide is formed, which is soluble form, and it can be excreted. You can see here, UDP is having phosphate group. Without phosphate, 2-UDP cannot be with glucuronate. Then this glucuronate cannot be donated to the bilirubin. If there is no 2-UDP glucuronate, bilirubin cannot be conjugated to bilirubin diglucuronide. So this bilirubin will be elevated. For the formation of 2-UDP glucuronate, you need a phosphate. Okay, phosphate groups in the liver. Where exactly it is occurring? In a liver. You can see here. In a liver, you need phosphate. If you have phosphate, 2-UDP glucuronate will be formed, and this 2-UDP glucuronate will convert this bilirubin into bilirubin diglucuronide. So conjugation process will occur and eventually it will get excreted by the body. Since phosphate, see here I told you phosphates are very much essential. That's why I put a star mark over here. But these phosphates are absent now, I am saying. Remember, these phosphates are absent. Why these phosphates are absent here? The phosphates are not there. The phosphates are depleted. Liver is short of phosphates. Why liver is short of phosphate? You can see here. You can easily understand. In the deficiency of galactose 1-phosphate uridyl transferase, what happens? Galactose can be converted to galactose 1-phosphate. Where exactly? In the cytosol of the liver cell. Galactose will be converted to galactose 1-phosphate, but this galactose cannot be converted to these two guys. Okay, so as soon as galactose enters into the liver, galactose utilizes phosphates, cytosol phosphates of the liver. See here, cytosol phosphates are there. See, these liver cytosol phosphates are getting utilized for the formation of galactose 1 phosphate formation. So whatever phosphates are there left in the cytosol of the liver cell are getting utilized by the formation of galactose 1 phosphate. Due to this reason, all the phosphates in cytosol will be depleted, and there is no phosphate left for the formation of 2-UDP glucuronate. Let me erase it for your clear understanding purpose. Yeah, there is no phosphate in a liver cytosol for the conversion of for the formation of 2-UDP glucuronate. So hence there is no 2-UDP glucuronate and there is no conjugation of bilirubin. If there is no conjugation, what happens? This unconjugated bilirubin will be what? This unconjugated bilirubin will be elevated. Okay, so you can see here in a clinical case, unconjugated bilirubin or indirect bilirubin is 14 mg dl. Okay, so bilirubin will be elevated and eventually what happens? It leads to jaundice. So since it is occurring inside the liver, you can say hepatic jaundice. Hepatic jaundice. See how galactose metabolism is correlated to the metabolism of heme. Due to the accumulation of galactose 1 phosphate, this galactose 1 phosphate accumulation leads to blockade in conjugation reaction of the bilirubin. So elevated levels of bilirubin and results jaundice. So here you can see, if there is no galactokinase, there is no galactose 1 phosphate related transferase, the cause is due to the deficiency of these two causes galactosemia, which means elevated levels of galactose in blood and elevated levels of galactose in urine also. Presence of galactose in urine. In urine, galactose is present. In blood also, galactose is present. So here you can see it. Let me let me answer for these 4 sub questions here under the clinical question. Now we will write here, what is the probable diagnosis and mention the biochemical defect. The probable diagnosis is this one, galactosemia. Yeah, the probable diagnosis is galactosemia. Okay, why galactosemia? Due to the deficiency of galactose 1 phosphate uridyl transferase. In shortly I am writing here because there is no space. Galactose 1 phosphate, this is one. Let me write it in a full form so that you can understand. Due to the deficiency of galactose 1 phosphate uridyl transferase. You can write in a full form if you get this question in exam. Okay, galactosemia due to galactose 1 phosphate uridyl transferase. On noting the above signs and symptoms, child's urine sample was tested for a particular compound. Mention the name of that compound. As I already told you, if the galactose is elevated, what happens? Galactosemia. The same galactose is present in urine also. If you test for reducing sugar, which is galactose, so galactose will be reduced for the Benedict's test. If you perform Benedict's test with this baby's urine sample, you will see galactose in the galactose will be positive. So on noting the above signs and symptoms, child urine sample was tested for a particular compound. What compound is it? Galactose only. So if the galactose metabolism is defected, means obviously galactose is present in the urine, which is galactose-urea. So you also mention the condition galactose-urea. And the third one is describe the metabolism of the above compound detail. So above compound is galactose. Okay, so galactose metabolism, which means you have to draw this whole pathway, galactose pathway. But remember, you need not to draw this whole heme degradation. Heme degradation is not required, but galactose metabolism you have to explain the whole pathway, and these deficiency of these two enzymes leads to classical galactosemia complications and everything you have to explain under this one. So I am writing here galactose metabolism you have to draw the whole pathway and also complications, enzyme defects and everything you have to write. Explain the reasons for the above signs. So what all the signs we have seen here? Yellow is discoloration of skin, we have seen discoloration within the eyes, we have seen bilateral cataract, hepatomegaly, everything we have seen. So all these see here. And also fasting hypoglycemia. Why baby is crying? Because there is no glucose inside. Because so babies can produce glucose from galactose. If there is no galactose metabolism, the metabolism is just blocking up to here, there is no formation of glucose to glycolysis and energy. Okay, fasting hypoglycemia. Why fasting hypoglycemia? Why fasting hypoglycemia? Because galactose 1 phosphatidylate transferase is absent or defective. Since this galactose 1 phosphate is not forming glucose 6 phosphate and results blockade in glycolysis. And also you all can explain why the reason behind the high levels of bilirubin, conjugated, unconjugated and total bilirubin, why all these are elevated, you can easily write. And also why it is discoloration in the eyes, within the eyes, and bilateral cataract due to Gelsitol. Gelsitol elevation leads to cataract, bilateral cataract in a baby. So all these are. Yellow is discoloration because jaundice. Yellowish discoloration is called jaundice due to blockade in conjugation of bilirubin. So this is all about this clinical question. In order to understand this clinical question, what you have to learn is, you must have to learn is these two pathways. The basics of heme degradation, and the basics of galactose metabolism. Galactose metabolisms are required to understand the whole clinical question. Always remember, while going through the clinical question, just underline the main important complications. So you may not find all the complications, you may not answer for all the clinical complications, but you can find a very important clinical finding like bilateral cataract, yellowish discoloration. Where can you see these two conditions? Mostly yellowish discoloration means definitely jaundice. So you can come to the confirmation. You can like these are all the clinical symptoms will make you get a conclusion on the diagnosis. So always try to highlight some important clinical complications that are unique in particular metabolism, so that you can easily answer for that. And also always be aware of normal reference values of all the biochemical metabolites and biochemical waste products, and everything you have to be aware of. Then only you can answer for the clinical questions. So I hope you understood this clinical question. If you understood clearly everything, please get subscribed to my channel, like it and share with your friends. If you have any queries, put your question in a comment box. Thank you very much. All the best for your exams.